sorted. Happy?
Cool.
Okay.
Yes.
Thanks.
Okay. Good morning, everyone. Today I will be presenting on the tolerance of apple rootstocks against Phytophthora root rot and white root rot. I'm an MSc student in plant pathology under the supervision of Prof. Adele McLeod. Phytophthora and white root rot are soil-borne diseases that attack the plant's root system. Phytophthora cactorum is the most important causal organism of Phytophthora collar, crown, and root rot. This pathogen leads to reduced root weight, stunting, and gradual tree decline, ultimately resulting in reduced yield. White root rot, on the other hand, is caused by Rosalinia necatrix and can be characterized by the formation of a white mycelial mat forming around the root system. This pathogen results in wilting and leaf necrosis, ultimately leading to the onset of rapid plant death. Rootstock choice can be considered as a management strategy for soil-borne diseases. Therefore, our study investigated the tolerance of certain Geneva and the industry standard rootstocks against the respective pathogens using local isolates. The trials were conducted under glasshouse conditions. In the Phytophthora trials, we evaluated root rot by mixing a sand bran inoculum of Phytophthora cactorum into the growing medium at planting. The trial was conducted over a period of 3 months, whereafter the total plant weight was measured and used for statistical analysis. We conducted 3 glasshouse trials in total. However, we had difficulty to induce disease symptoms in 2 of these trials, and therefore I will only present the preliminary data obtained from the 1 trial with successful inoculation. This slide shows all the rootstocks that were tested in our study, and I'm highlighting rootstocks that were significantly more tolerant within a vigor class compared to the other rootstocks that were tested. The more tolerant rootstocks included G222 and G757 in the dwarfing class, G210 in the semi-vigorous class, and M793 and G228 in the vigorous class. The industry standards M9, M7, and MM109 were all found to be susceptible in this trial. Since this is preliminary data, it is important for the trial to be repeated to confirm the results obtained. Moving on to the Rosalinia trial, we used a bamboo stick inoculum, which involved inserting mycelium-colonized bamboo sticks of 2 different inoculum dosages, either a 6 or 12 sticks that were inserted into an individual pot. For the duration of the trial, disease was evaluated using a disease rating scale, ranging from a healthy to a completely dead plant. The days until plant death was used to determine the tolerance of the different rootstocks. Preliminary results of 2 Rosalinia trials showed that all rootstocks are susceptible to this pathogen. It is only in the dwarfing class where G202 and G757 were found to be slightly less susceptible, and this was also at a lower inoculum dosage. This graph illustrates the effect of inoculum dosage on disease severity of the pathogen, and I'm highlighting G202 and G757 to show the delayed onset in plant death that was observed at the lower inoculum dosage, indicated by the maroon bars. Across all rootstocks and across all vigor classes tested, the similar trend was observed where at a high inoculum dosage, indicated in the green bars, more rapid onset of plant death occurred. So to conclude, through the preliminary results obtained in our Phytophthora trial, we could identify rootstocks within each of the vigor classes that were significantly more tolerant to Phytophthora cactorum compared to the other rootstocks tested. In the Rosalinia trial, all rootstocks were found to be susceptible. And because this pathogen is so severe and leading to total plant death, it is important to note that a less susceptible rootstock cannot be considered as a viable management strategy, and management can only be achieved through a rootstock with complete tolerance against Rosalinia necatrix. Lastly, this study also highlighted the important effect of inoculum dosage on the disease severity of the pathogen. This will have implications on the results of orchard trials, since the quantity of inoculum that a tree is exposed to will directly influence the results of such a trial. And with this, I would like to thank you.